Well, welcome to this discussion on how important the subject of biochemistry is in food processing. My name is Paul Singh. I'm a professor at University of California in Davis, and I was here in Guangzhou and decided that I will take part in this introductory comments related to the importance of biochemistry in the field of food science. As you know, that foods have got variety of components, whether it is proteins or carbohydrates or lipids, and then there are other components such as enzymes. And we are very often interested in how these different compounds interact with each other, because that is what allows us to perhaps modify a food. Those are the kind of changes that take place when food processing is carried out. So understanding the underlying mechanisms, the mechanisms of those reactions, understanding the rate kinetics, that is how fast a reaction is taking place, is very important, because that helps us to design food processes. So I'm glad that you are in this course and you are going to study and really get a good understanding of a variety of chemical and biochemical reactions in depth. So that you will be able to design new food processes, you'll be able to create novel foods in your own careers. So let me give you an example from some work that we have been doing at University of California, Davis in my laboratory. And this will show you how important biochemistry is in carrying out a certain research project, as well as trying to get better understanding. So this project that we have been working on for the last 10 years relates to what happens to food after we eat that food. Because inside our human body, there are a number of biochemical reactions that take place. So we have been trying to understand those types of reactions, trying to understand and measure what are some of the kinetics, the rates of those reactions. So if we look at this slide, you will notice that the first place where the food comes into interaction with the human body is in the mouth. And in the mouth, as you chew a food, you actually are mixing that food with an enzyme. And those enzymes are coming from saliva. So that is where many of the enzymatic processes that are related to breakdown of carbohydrates are important. And then as the food moves through the esophagus and then goes into the stomach, now you have a situation where the food that you have chewed is now coming into contact with acid, and also additional enzymes. Those enzymes are pepsin. Pepsin is an enzyme that breaks down proteins, and also certain gastric lipase. Lipase is the enzyme that breaks lipids. So in the stomach, the food mixes with the acid, which is being released from the walls of the stomach, and also mixes with the enzymes. So again, there is food breakdown, further breakdown, due to the action of acids and enzymes. Now, that is the subject of biochemistry, because those biochemical reactions that are taking place, we want to understand not only the types of reactions, but also the kinetics of those reactions. From the stomach, the food moves into the intestines. Again, in the intestines, there are further biochemical reactions that take place, where further breakdown of food occurs. And enzymatic hydrolysis occurs in the intestines. From there, the food moves to large intestines. In large intestines, there is a fermentation process that takes place. So as you see, there are a number of biochemical reactions that are going on right now in everybody's human body. And for us as food scientists and food engineers, we must get a good understanding of those reactions and the kinetics of those reactions in the human body. So that is the study that we have been doing. So if we look at the stomach, this is one part of the body that we have studied in more detail. Here in this slide, we see that we mix the food with gastric juice, which is mainly hydrochloric acid. That is what the body secretes from the walls of the stomach into the food. So there is hydrochloric acid, there is mucus, there is pepsin. Again, if you recall, I mentioned that pepsin is an enzyme that breaks down the proteins into smaller polypeptides. And then also a certain amount of salt and gastric lipase. So if we look further, we notice that here is a slide of the human stomach and the inside walls of that human stomach. There is a part of the stomach we call the fundus area, which is on the top of the stomach. There is the general body as well as the antrum. Antrum is the lower part of the stomach. That is where most of the food that we eat breaks down due to those biochemical reactions. So in this slide, we see that depending on what type of food you are eating, they will have different breakdown mechanisms. This is a slide that shows that if you take a piece of carrot and you cook that carrot for different time periods, let's say you have raw carrot, or you cook it for 2 minutes or for 6 minutes, and then if we look at how that piece of carrot will break down in gastric juice along with the enzymes, how long would it take for it to break down and how would it break down? That is important because large particles have to become smaller particles so that later on the molecules that you are interested in, the nutritional molecules, can release into the bloodstream. So here in this slide we see that the raw carrot, which is the graph with the red color, the breakdown is much slower than if you cook that carrot. Now I'm sure intuitionally you will also think of the same, that if you cook something, it gets soft and it will break down faster. Except now here in this graph, we have actual kinetics, the rate at which it breaks down. And that information is important in designing food processes. Here is in this slide, we look at a mechanism of how this breakdown takes place. So in this case, we see that there can be a situation where the acid diffuses into the food. So the diffusional process becomes very important. Again, this is going to control the biochemical reaction. As the acid diffuses, it breaks down the structure so that the surrounding acid then causes erosion of the food from the surface. And this is how the particles become smaller and smaller. As you can see in this slide, this is a piece of carrot again. And if you can notice that there is a slight bluish tinge, blue color tinge on the surrounding. And that is the part where the acid has diffused into that piece of carrot. And in this slide, you see how the breakdown of the pectin layers have taken place because of acid. Again, these are biochemical reactions taking place in the food product that allows that breakdown. We can, with this type of knowledge, decide on how different types of foods, for example, foods like nuts, like almonds, or food like an apple slice, how they are going to break down inside the food and we can use that information then in designing new food products. Let me share with you briefly a trial that we did. This is what we call in vivo trial, where we use either humans or animals to conduct these types of studies. Here again, you will get an idea about how biochemistry, or the role of biochemistry in food breakdown inside the gastrointestinal tract. So one can do in vivo trials with different animals like rats or pigs. Our studies were done with pigs that were euthanized after they were fed a certain diet. So just to give you an idea of how this study was done and why we selected pigs, because the pigs have very similar upper GI tract as human beings. So the studies we do with this animal helps us to understand what happens in a human GI tract. So in this case, in this study, we were looking at the rate of breakdown. We were looking at how food processing, the role it plays in these breakdown mechanisms because of biochemical reactions such as hydrolysis, enzymatic hydrolysis, and so on. And then also we were looking at mixing. How does, for example, acid mix with food? And we looked at two different foods, two different types of food categories. One was where we compared brown rice with white rice. As you know, brown rice has got the bran layer intact, whereas in white rice, you remove the bran layer. In case the other product we looked at was almonds. So we considered raw almonds and roasted almonds. Again, we are trying to understand how different food processing methods affect foods and how they break down inside the human body. So for the trials with pigs, you have to conduct an experiment with sufficient number of samples. You have to do it correctly as far as statistics is concerned. So in our study, we had to consider about 150 pigs who were acclimatized for a certain period, and their diet was prepared. The diet was the cooked rice, brown rice and white rice, and raw almonds and roasted almonds. And they were then prepared for feeding at a certain time. This slide shows you the stomach of a pig, which is again very similar to a human stomach. And these diets were fed to these pigs, and then they were euthanized at different times. For example, after feeding them, a certain number were euthanized at 20 minutes, 20 minutes, 60 minutes, 120 minutes, and so on. And then we could draw samples of pH to measure the acidity. We measure the moisture content, textural properties, rheological properties. So we can do all those types of measurements. And we also looked at the amount of protein and amount of starch, again looking at the biochemical reactions. This slide shows you that after 20 minutes of digestion, we can compare brown rice and white rice. You are looking at how the food is inside the pig's stomach that has been euthanized and then removed and opened to get the samples. You see that the color is blue. That is because we want to do imaging. That's why it's a blue color. But if you notice here, in case of brown rice, in the lower part, that is where you have the bran layer coming off the brown rice. So this is a process taking place inside the stomach. Initially, when you ate, or when these pigs ate this product, the bran layer was intact with the rice. But when it went through the digestion, as a result of the acids and enzymes, that bran layer came off the rice kernel. On the other side, you see in white rice, now there is more breakdown towards the lower part, and as you can see, the size of the rice kernel getting smaller. We can look at again after 2 hours of digestion, you see more of the breakdown and separation of the bran layer, and this is after 8 hours. So very clearly, just from these pictures, you can see that the biochemical reactions that are taking place are impacting the starch as well as the protein that is in the bran layer. And the similar type of results we can see with almonds. Again, the pieces of almonds are being influenced by the enzymes and the acid. One last thing that I will show you here is how the pH level varies inside the stomach, because that is what is impacting the biochemical reactions. And here, for example, we have samples drawn from various parts of the stomach. And you can see in this slide, on the top you have the brown rice that was in the stomach, and the lower one is for white rice. The red color shows that pH is very low, like a pH of 1. On the blue side is where pH is 7, so it is a high pH. And what becomes quite obvious from looking at this, that there is a certain amount of mixing that takes place in the stomach. But in case of white rice, which was influenced by the biochemical reactions, you have a complete low pH towards the latter part of the digestion process. So at 480 minutes, you see it's all red. Whereas in case of brown rice, which is slow in breaking down, where the biochemical reaction takes place at a slower kinetics, you still have regions where the pH is not that low. So you might ask, well, how does that affect digestion? Well, many of the enzymes have an ideal pH where they are most effective. So if there is not the ideal pH present, then you don't have sufficient breakdown. So these are the things that you will learn in this course. As you study different parts, for example, proteins, you learn about lipids, or you learn about starch, you will see how many of these reactions take place under what conditions. And with that knowledge, you will be able to then analyze what I just showed you from a research paper and get a better understanding of how what happens just in the human body, and how in food processing we can create novel foods for novel purposes, where we may want a particular nutrient to be available in the human body for healthy foods. So I wish you the best in taking this course, and I hope that you learn many of these aspects of biochemistry, and I can assure you that with that knowledge, you will be able to make some major advances as well as impact the future development of the food industry. So, thank you.